Hello students, today we are going to study about the Mendelian crosses or the famous Mendelian crosses. Okay, so there are specifically two types of Mendelian crosses. One is monohybrid, one is dihybrid. Okay, monohybrid cross and dihybrid cross. So initially we are going to talk about the monohybrid cross. Okay, so what do you think? What is monohybrid? Okay, mono means one. Okay, mono means one. That means only one trait was taken. Trait means characteristic. Trait means the characteristic. And here, what was the characteristic which was taken by Mendel? Okay, can you tell me? Yes, that was the height of pea plant. Height of pea plant, because that was the only thing that which was available for him. Okay, as you know, now there are so many things which are available. So we are going to talk about the height of pea plant. And what he did, he took the one pea plant of the pure tall trait. Means there was nothing like one was in the whole pea plant, one pea plant was tall, one was dwarf. No, if he took a whole plant as a tall and one as dwarf. Means the characteristics were pure, not mixed. Means the alleles, there are pair of alleles in a gene. So he took pure parents. So what he actually did, now we are going to cross over it. Okay, he took parent generation as big T only. Okay, the tall one and the dwarf one, big T and big T for tall. Wait a minute. So the big T and big T for tall and small t and small t for dwarf. Okay, so what he did, he make the gametes. What he did, he separated the gametes. Specifically what we can say, okay, he separated the gametes means the genes, and there are pairs of alleles in one gene. Okay, now you have to understand this. Please do not forget this. There are a pair of alleles. Okay, now we are going to the simplest crossing, like big T with small t, and that big T with another small t, and another big T with the small t, and that last big T with small t. Now you can see all are tall plants. Okay, now we are going to make a Punnett square. This was the regular one crossing, but if your teacher is asking, you can make a Punnett square also. Okay, so you can cross this T to this T is big T, and this T to big T and small t, and big T and small t to big T and small t. You can see in both crossings the result is same. You can do whatever you want. Okay, now we are going to study the which one is the dominant gene. Okay, dominant gene here is the tall one. Dominant gene is the tall one. As you can see, this is the F1 generation. This is what? A F1 generation. Why? Because it's come from a parent he was experimenting on. Okay, why we are saying the tall ones genes are the like dominant one? Why? Because all the plant in F1 generation come as tall. Okay, now we are going to make the second one, means the second generation. Okay, so we are going to make the second generation. And for the second generation, the Mendel took the first generation plants as the parent one. Okay, he took the F1 generation as the parent one here to get the F2 generation. You can see from where I have taken the big T small t and big T small t. Okay, you can see in this Punnett square above. Okay, so after making the gametes, okay, so now after making the gametes, we are going to do the similar crossing, like big T with big T, that big T with small t, and small t with another big T, and small t with a small t. Okay, okay, that's what the cross happened. Now if you want a Punnett square, what a Punnett square, because majorly the teacher will ask. Okay, now we are going to make a Punnett square here. So the Punnett square will be like similar one, big T with big T, big T with the another small t, this small t with big T, the big T will be, okay, will be the first. Okay, you cannot write small t with in the first and then big T in the second. No, because big T is the dominant gene. Okay, so this is the second generation. This is what we get as the second generation. Now we are going to write what? The phenotypic and genotypic ratios. Okay, so here you can see how many there are. How many tall plants? There are 3 tall plants. As you can see, in 3 genes you can see the dominant tall gene. Okay, but in the 4th one you can see there is no trace of dominant gene. So one pea plant will be dwarf in second generation. So what you can see in the physical form, because you cannot see the genes in real, okay, in front of your eyes. So what you can see is the phenotypic ratio, like how many tall and how many short you can see the plants. So there are 3 tall plants and 1 dwarf plants. Okay, so the ratio phenotypic will be, means the physical one will be this 3:1. And the other thing we are going to talk about is the genotypic ratio. Is what? The genotypic ratio. Genotypic ratio means how many types of gene you can see here. Okay, you can see the big T, then big T, small t, big T, small t, means there are 1, 2 and 3. Okay, from F2 generation only you can give the phenotypic and genotypic ratio, not from the F1 generation. Okay, because there are only tall plants and only one type of genes.